Mash'an joined our lab almost 6 years old, and he finished the master degree from our lab, and now he's finishing the PhD requirement. Mash'an is one of the students who I ever see student like him. He's very sincere and concerned about his work, and he almost hanged with the research in every time. So anytime we direct him to any research, he do it. And today he published 3 papers from his PhD, and he has another paper from different work. So today when I submit him for the PhD, I think he's qualified and he can do independent research without even postgraduate work. I would like to welcome Mash'an, and I would like to appreciate the department for the help for me and for Mash'an. Welcome, Mash'an. Start your presentation.
Thank you, Dr. Othman, and I would like to also thank you and all the committee members and the people who they are with us today. And if you allow me, I will start my presentation now. Can everybody see the screen now? بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. It's a great pleasure to me to present my thesis today on a presentation. This presentation will be titled under the title of The Role of Fatty Acid Synthase and Carnitine Palmitoyltransferase, one pathway in molecular subtypes of breast cancer as a potential targeted therapy. This presentation will include introduction, hypothesis, aim of the work, material and method, statistical analysis, results, and conclusion. I will start with a brief introduction of the cancer. A cancer is abnormal growth of the cells which tend to proliferate in uncontrolled way and in some cases lead to metastasis. Regardless of all of the improvements made over the years in controlling the cancer and developing treatment for the cancer, cancer is still the second leading cause of death in the world after cardiovascular disease. As you can see here in the chart, there is an increase in the incidence and the mortality for the cancer over the years, and we still have, and there is still an anticipation that in 2030, maybe we will have an increase in the incidence of cancer cases up to 21 million, and the cancer-related death may reach to 13 million. The most top 10 cancer in both male and female are the highly incidence cancer is the lung cancer and breast cancer, and the highly mortality cancer is because of lung cancer and colorectal cancer. If we divide these things into the male and the female, the highly incidence cancer in male is the lung cancer and the prostate cancer, and the highly mortality is the lung cancer and liver cancer. Whereas in the female, the highly incidence and the highly mortality is the breast cancer. There is a different risk factor related to the link between the cancer, and these factors like aging, carcinogenesis, stress, tobacco smoking, food additives, chemicals, and many other things. Usually for diagnostic purposes, the classification of tumor molecular subtypes is based on the level of expression of relative receptors by immunohistochemistry staining, as well as the level of expression profile. But basically, we can classify the cancer into the epithelial tumor, non-epithelial tumor, and unclassified tumor. For example, if the epithelial tumor is the most of the malignant carcinomas cases, or the adenocarcinomas like cancers of the breast, lung, or prostate. Breast cancer is the leading cause of cancer-related death in more than 100 countries. worldwide about 2.1 million new cases of breast cancer are diagnosed in 2018. And annual breast cancer death rate will exceed 600,000 every year. The researcher attempted to evaluate the 5-year survival rate for breast cancer patients and found it is around 89%. And this number dropped by 25% if metastasis occur. Several studies have identified the link between breast cancer and hereditary. And they estimated that approximately from 5 to 10% of the breast cancer cases has some relation with some famous genes related to the mutation like BRCA1 and BRCA2. What is the situation here in Saudi Arabia? According to the most recent National Cancer Registry in Saudi Arabia, breast cancer is at the top and the most frequent tumor among all malignancy seen in Saudi females, and it accounts for more than 30% of all female cancers. Breast cancer in young Saudi women is a crucial problem with the proportion of young age onset and the breast cancer much higher than in Western countries. Data on female with invasive breast carcinoma reported from different regions in Saudi Arabia, and most of patients are diagnosed in the age group from 40 to 55 for 54 years. And as you can see here in the chart, the incidence increased every year. In 2000, we had around 10 million. In 2010, we had 27 million.4. And according to the last report, 2018, it's up to more than 30 million. There is a different risk factor related to the breast cancer. Of sure, the hereditary may represent 15% of the cases. And also there is a relation with the personal factors, including the hormonal status, relapse, hormonal replacement therapy, early menarche, few numbers of children, but also high breast density, and the lifestyle included in this as one of the risk factors, like weight gain, high calorie intake, obesity, and others. The issue with the breast cancer classification is that the breast cancer is a highly heterogeneous disease and composed of different subtypes, which can be determined by gene expression pattern or by the immunohistochemistry. Identification of the subtype is of a great value because each subtype has a distinct biological and clinical feature which represent differentially to different treatment protocol and prognosis. We can classify the breast cancer according to the histopathological characteristic like morphology, tumor size, tumor grade, receptor status, and nodular involvement. But also we can classify the breast cancer depending on its origin. We can divide the breast cancer into in situ carcinoma, which is the mostly non-aggressive phenotype, and invasive carcinomas, which is the aggressive subtypes, and this aggressive subtypes can be divided according to the luminal subtype as a luminal A, luminal B, triple negative, or HER2 subtypes. If we divide the breast cancer only in the molecular subtypes, we have 4 major subtypes, luminal A, and representing more than 50% of the case. And this subtypes has estrogen positive, progesterone positive, and HER2 negative. And we have also luminal B, which represent 15% of the cases and has representing the estrogen positive, progesterone positive, and HER2 positive. Triple negative subtypes has negative expression for estrogen, progesterone, and HER2. The last subtype is the HER2 subtypes, which are represented by estrogen negative, progesterone negative, and HER2 positive. I will give us short information about each subtype here. For example, the luminal A subtype is characterized by high level of estrogen or progesterone positive tumor with a negative for HER2. Luminal A is the most common subtype of breast cancer, represent from 50 to 60% of the case, and has in general good prognosis profile and lower relapse. The treatment for tumor with the luminal A subtypes mainly based on the hormonal therapy, and the primary drug used is for the either estrogen blocker like tamoxifen or aromatase inhibitor like anastrozole. The other one is the luminal B. The luminal B is defined as estrogen, progesterone, and HER2 positive. This subtype of breast cancer represents from 15 to 20%. It is one of the mostly aggressive phenotype and characterized by high proliferative index and histological grade. Also, there is a high recurrence rate with low survival rate after relapse. It's more important to mention that the using of endocrine therapy with the luminal A breast cancer was relatively insensitive in compared to the luminal A breast cancer with poor outcomes. The other type is the HER2, and it stands for Human Epidermal Growth Factor Receptor 2. It's one of the family of the tyrosine kinase receptor, which is a transmembrane receptor protein. This tumor is represented around from 50 to 20% of the cases and has aggressive clinical and biological behavior. Also, this type is characterized by high expression of the HER2 gene, and more than 40% of the cases have a mutation in p53. Mostly the treatment for this subtype is by using the trastuzumab, which is a monoclonal antibody targeting the extracellular domain of the HER2 protein, and this will lead to improving the patient outcome. The last subtype is the basal-like subtype or triple-negative subtypes, and this subtype does not express estrogen or progesterone or HER2, and hence referred as a triple-negative. And this type of breast cancer represents from 8 to 37 of the cases, and this wide range, it's different because there is a wide variation in the literature and from country to country. And all breast cancer cases associated with a poor tuberal formation and with high rate of metastasis to the brain and the lung. In fact, the patient with triple negative cannot have a benefit from using of endocrine replacement therapy or HER2 antagonist like trastuzumab, and the primary treatment for those patients is using the cytotoxic chemotherapy like cyclophosphamide, ifosfamide, carboplatin, or doxorubicin. And this subtypes have the worst outcome compared to the patient with the other subtypes. So what are the different subtypes that we have it in Saudi Arabia? We are not much far away from the world. We found in the literature 3 different big studies investigating those different subtypes, one from the eastern region, another one from the central region, and the last one from the western region. In the central region, the most abundant type is the luminal A, and it represents more than 58% of the case. In the western region, the luminal A also represents 47% of the case, whereas actually inversely, we have different data from the eastern region, whereas the majority of the cases is the HER2 subtypes, which is different than the other countries in the other region in Saudi Arabia or the western countries. Let's talk about a little bit about the cancer metabolism. Metabolic reprogramming is one of the newly accepted hallmark of cancer, and it is well known that the cancer cells are highly growing cells, and accordingly they need a high metabolic reprogramming system to maintain their needs of growth. Several studies demonstrated that the de novo fatty acid synthesis is the highly induced in different types of cancer, and this induction can be used for providing both raw nutrient material for synthesizing the new cell membrane to give more ability for the cell for proliferation, and also to protect the cells against the oxidative stress. Fatty acids are essential in energy storage, and also for modification of post-translation and maintaining the function of some regulating protein. In mammals, most of the tissue use the circulation to obtain their need of fatty acid, whereas some other tissue are synthesized their own need of fatty acid through the de novo fatty acid synthesis pathway. Fatty acid synthesis pathway, it's require the presence of fatty acid synthase. Fatty acid synthase is a lipogenic enzyme which is responsible for the synthesis of long-chain fatty acid from acetyl-CoA, malonyl-CoA, and NADPH. FASN is overexpressed in most of the human malignancy and therefore is considered as an ideal therapeutic target. Consistent with this theory, several studies have clearly demonstrated that there is a high level of fatty acid synthase expressed in most human tumors, including breast, prostate, stomach, skin, and other types. In normal cells, the indigenous synthesis of long-chain fatty acid is minimal because diet supply most of the fatty acid. In vivo and in vitro study reported that the treatment of tumor cells with a pharmacological inhibitor of FASN lead to apoptosis of breast tumor cells, and high level of malonyl-CoA, which continue to be generated by acetyl-CoA carboxylase, which is the rate-limiting enzyme in the fatty acid synthase pathway. In the breast cancer cells, it was reported that there is inhibition of FASN by FASN small interference RNA is associated with apoptosis. So what is the situation from the literature? If we have any FASN inhibitor, this will lead to inhibition of fatty acid synthase, and this inhibition will lead to accumulation of the malonyl-CoA because the malonyl-CoA is the substrate of fats. This accumulation of malonyl-CoA lead to inhibition of CBT1 because malonyl-CoA is the normal physiological inhibitor of CBT1. By inhibiting of the CBT1, we will inhibit the high amount of fatty acid to transfer from the cytosol into the mitochondrial for beta oxidation, and this high amount of fatty acid that will be accumulated in the cytosol will lead to formation of high amount of ceramide, and this ceramide will lead to activation of the apoptosis in different cycles. At the same time, this inhibition will lead to accumulation of the NADPH and the formation of NADPH oxidase and high amount of the active oxygen species. At the same amount, high amount of the inhibition of the palmitate production will lead to inhibition in the phospholipid formation. So let's talk about these genes in a little bit. I'll start with the CBT1A. CBT1A is the carnitine palmitoyltransferase 1. It's present in mostly in the liver, kidney, duodenal, and small intestine. It's available at the outer mitochondrial membrane, and several studies confirm there is a relation between the overexpression of the CBT1A and the tumor progression in several types of cancer. Also, it's been reported that the CBT1 inhibition is associated with the apoptosis and suppression of the proliferation of cancer cells. Also, the use of etomoxir as a CBT1 blocker, it's one of the chemical inhibitor for CBT1. It's been tested on prostate cancer, and this leads to increase in the viability of the prostate cell lines. The other gene is the acetyl-CoA carboxylase. It's a rate-limiting enzyme in the synthesis of fatty acid by converting the acetyl-CoA into malonyl-CoA in the presence of the ATP. In fact, several studies demonstrated that the ACC1 is upregulated in different cancer cells, and like the prostate cancer, liver cancer, and breast cancer. Use of ATOFA, one of the chemical inhibitor of ACC1 and ACC2, lead to growth arrest and the cytotoxicity in the prostate cancer cells and in the cells in vitro. Also, it's been used with the lung and colon cancer cell, and this lead to reduction in the fatty acid synthesis and cell death. The other gene is the malonyl-CoA decarboxylase. This enzyme is responsible to decarboxylate the malonyl-CoA to give acetyl-CoA. The MCD messenger RNA and the protein are strongly expressed in the heart, liver, kidney, and skeletal muscles. In the cytosol, both ACC1 and MCD are responsible for controlling the level of malonyl-CoA. Therefore, MCD can regulate the fatty acid oxidation indirectly by controlling the level of malonyl-CoA. So either using of small interference RNA against MCD or using the chemical inhibitor like MBA as an inhibitor for MCD, this will lead to increase in the level of malonyl-CoA and lead to cytotoxicity in breast cancer cell, but not the fibroblast. So let's imagine the situation in the normal cells. As you can see here, in the normal cells, we have normal expression level of the malonyl-CoA and MCD, malonyl-CoA decarboxylase, and ACC1. Accordingly, we have a stable level of malonyl-CoA and acetyl-CoA, but we have a very minimal expression of fatty acid synthase because most of the fatty acids, palmitate or fatty acid coming from the dietary supply and can pass easily from the cytosol into the mitochondria through the CBT1. So what could happen if we have what is the situation in the malignant cells? According to the literature, the malignant cells represent high expression of fatty acid synthase. This high expression needed to produce high amount of the palmitase, as we say that the malignant cells need this palmitate to produce cell membrane, post-translation modification for proteins, and many other things. And for use of this fatty acid synthesis, we need high amount of acetyl-CoA and malonyl-CoA for producing the palmitate. But what could happen if we have one of the FASN inhibitor or CBT1 inhibitor? Inhibition of FAST, this will lead to accumulation of the acetyl-CoA and also accumulation of the malonyl-CoA. Accumulated malonyl-CoA will lead to inhibition of the carnitine palmitoyltransferase 1 receptor by malonyl-CoA, and this inhibition will lead to accumulation of the palmitate that will come from the circulation. And the circulation still support the cells with the palmitate, but there is no way for the palmitate to go for the mitochondria. It start to accumulate more and more, and by the presence of the sphingosine and ceramide synthase, we will have much more amount of the ceramide. The ceramide will initiate the apoptosis in the cells. So I want to go just shed some light on some related research in the same topic. We can see here using an immunohistochemistry study, there is a recent study examined the FAST expression in around 29 triple negative block and reported that the FASN staining was observed in all tumor samples with the high positivity in more than 30% of them. The author showed that the therapeutic benefit of FASN inhibitor in sensitive and chemo-resistant triple negative preclinical model, as well as the strong synergistic anti-tumor effect of FASN inhibitor in combination with cetuximab. A limitation of this study is that the author did not investigate FASN expression in non-triple negative phenotype. Sorry. If there is any differential, and the FASN expression was not investigated on scoring non-quantitative level, and the FASN not studied in normal non-malignant breast tissues. The other study investigated the immunohistochemistry staining of FASN in more than around 600 cases of invasive breast cancer and found that the FASN expression was positive in more than 70 cases and was inversely correlated with the histological grade and positively correlated with the expression of estrogen, progesterone, and HER2. But again, the extent and the differential FASN expression in non-triple negative as compared to triple negative are not investigated. Other study from Korea investigated the expression of FASN and CBT1 in more than 400 patients with different breast cancer subtype, also by using immunohistochemistry, and the author found that the CBT1A and FASN expression was higher in all non-triple negative subtypes, with higher in HER2 tumors and lower in triple negative, and they concluded that the lipid metabolism related protein are differentially expressed in different subtypes of breast cancer. So that differential expression of FASN in different subtypes of breast cancer might explain the differential sensitivity or resistance of breast cancer to the currently available chemotherapeutic treatment protocol. And in these previous studies, the expression of FASN and related genes was not investigated, was investigated on scoring and non-quantitative level and not studied in non-malignant breast tissues. Moreover, the molecular mechanism of cytotoxicity whereby the modulators of FASN and related gene are not fully investigated in breast cancer subtypes. Accordingly, the current study hypothesized that FASN and CBT1 and their related genes are differentially expressed in breast cancer subtypes, and that genetic and pharmacological modulator of these genes might help in design tailored therapy, which is a trend in clinical practice these days. Now, if you allow me, I will shed some light about the eugenol, because we will use eugenol in this study as one of the pharmacological inhibitor for fatty acid synthesis or oxidation. Eugenol is a natural phenolic compound available in honey and essential oil or clove. It have a different use in medication, can be used for treat digestive disorder, antiseptic, analgesic, anti-inflammatory effect, has some antimicrobial property, also antifungal properties. Eugenol has several anti-cancer property as well, and it's been shown in cancer and prostate cancer and liver cancer as well, and breast cancer. In 2007, there is a study investigated the effect of eugenol on cellular energy metabolism, and the author found that the eugenol produced a dose-dependent selective cytotoxicity toward MCF10A RAS transfected cells, but exhibited no apparent cytotoxicity to MCF10A cells. And this effect was mediated mainly through the inhibition of ATP generation and CBT1C and HRAS transfected cell, not MCF10 cells. Other study in 2017 described the ability of clove extract as a natural fatty acid synthesis inhibitor and to prevent the obesity in mouse model. And in this study, they were screened the alcohol extract of clove as a FASN inhibitor in mammalian cells and tissue. Interestingly, they found that the alcohol extract of clove could suppress FASN expression and reduce palmitic acid body weight in high-fat diet mouse model. So up to date in the literature, we could not find any study investigate the effect of eugenol on gene engaged on lipogenesis and fatty acid oxidation in different breast cancer cell lines and its relation to the anti-cancer activity of eugenol. Therefore, the current study has been initiated to investigate whether the eugenol mediates its anti-cancer properties by modulating the gene involved in lipid metabolism in HER2 subtypes and in triple-negative subtypes. So what is the aim of this work? The aim of this study is to examine the effect of modulation of FASN and CBT1 pathways in different breast cancer molecular subtypes as a potential targeted therapy, which will be achieved as follows. First, to detect the differential expression of FASN, CBT1A, and related genes in breast cancer cell lines with different molecular subtypes, aiming at identifying molecular drivers that can be therapeutically targeted. Also, to clarify the molecular mechanisms whereby the pharmacological, such as eugenol, and the genetical modulator of FASN, CBT1, and their related genes contribute to tumor cell cytotoxicity in different subtypes of breast cancer cell lines. Now I want to shift for the material and methods. Sorry. In this study, we use 4 different breast cancer subtypes. We use the human breast epithelial, which is a normal human epithelial breast cancer cells. It's MCF10A, and we use laminal A presented by MCF7, HER2 subtypes represented by SKBR3, and triple-negative subtype presented by MDA MB231. We use different technique to achieve our goal. First, we use the RT-PCR for differential messenger RNA to detect the differential messenger RNA expression of FASN, CBT1A, ACC, and MCD in different breast cancer cell lines according to the known protocol. This is the list of Tachman primer being used in this study. Also, we use Western blot analysis for detection of protein expression of FASN, CBT1A, ACC, and MCD in different breast cancer cell line according to the known protocol. Here is the list of the primary and secondary antibody being used in this study. Also, we use small interference RNA for transfecting the cells with our target to transfect our target genes ACC, MCD, CBT1, and FASN. And we use the lipofectamine 2000 according to the company protocol, and we incubate the cells for 48 hours. After that, we analyze the transfection efficiency by RT-PCR. We accept the transfection efficiency if it's more than 50%. Here is the list of the small interference RNA being used in this study. Also, we use the MTT assay to evaluate the cell viability, and we detect the color by using the ELISA at 450 nanometer absorbance by I forget the machine name. Anyway, also we use the RTCA system, real-time cell electronic sensing system, to detect the cell proliferation analysis. We detect real-time cell proliferation for 72 hours for SKBR3 cells and MDA231. To detect the apoptotic activity for our small interference RNA and our pharmacological inhibitor, we used the annexin 5 assay, and we used the Muse system, which is a small flow cytometry system. Also, we use in our work the scratch wound healing procedure or assay to detect the ability of phegenol to inhibit cell migration. Statistical analysis. Data were expressed as a mean plus or minus standard error of the mean, and the difference between obtained values where when we have a two group of comparison, we use one unpaired student t-test, and when we have more than two group, we use one-way analysis of variance followed by Tucker-Kramer, and the p-value of 0.05 or less were taken as a criterion for statistically significant difference. I will shift now for the results. Data presented in the current study have demonstrated that the messenger RNA expression of FASN, CBT1A, ACC, and MCD are differentially expressed in all breast cancer cell lines under investigation compared to the normal human mammary epithelial cells. This is the messenger RNA expression data. As you can see, we have a different expression level upon the different cell lines for each different genes. We confirm the data that we have it from the messenger RNA by Western blot analysis for protein, and the data here, alhamdulillah, show a similar result from the messenger RNA. According to that, the current study has selected SKBR3 breast cancer cell line in subsequent experiment to target the FAS. As you can see, the messenger RNA data and the protein expression data shows that the the FASN are highly expressed in SKBR3 cells. Also, the SKBR3 cells used in subsequent experiment to target CBT1. Accordingly, as you can see here, that the CBT1 are high in SKBR3 and MDA in the messenger RNA, but when we go for the protein expression data, the high expression available in SKBR3 cells. So one can anticipate that the both fatty acid synthesis and oxidation are very active and essential for survival of HER2 subtypes. Also, we think this result suggests that the breast cancer patient with the HER2 subtypes will benefit more from FASN and CBT1 inhibitor than luminal A and triple negative subtypes. Also, this study has selected MDA231 breast cancer in subsequent experiment to target the ACC1. As you can see from the messenger RNA and protein expression data, the MDA represent high expression of ACC. In the current study also, we use the messenger RNA and the protein expression of MCD and are significantly expressed in all breast cancer cell line under investigation compared to the normal human mammary epithelial. Interestingly, MCF7 breast cancer cell line show the greatest MCD messenger RNA and protein expression, followed by MDA, then SKBR3 cell lines. So, since the FAS gene showed the highest expression in SKBR3 cells, therefore, FASN was knocked out in SKBR3 cells transfected with the either small interference RNA or control scrambled small interference RNA. And incubation of the SKBR3 cells with a FASN small interference RNA resulted in around 60% decrease in the FASN messenger RNA expression. As a consequence of this inhibition, the current study measured the messenger RNA expression of CBT1A, ACC, and MCD using RT-PCR to explore the possible effect of this inhibition on these genes. As we can see in the figure, FASN downregulation resulted in a significant 30% decrease in ACC1 messenger RNA and non-significant changes in the messenger RNA expression of CBT1 or MCD. To determine the effect of using FASN small interference RNA on the viable cells and apoptosis profile, the SKBR3 breast cancer cell lines in the SKBR3 cell lines, we use the annexin 5 and the flow cytometry analysis. As you can see in the flow picture, the treatment of the cells with the small interference RNA for 48 hours lead to apoptosis in compared to the normal control. Usually, you will find in this picture and all the coming picture, on the right side the treated cell and on the left side the control cell. Also, to explore the effect of using CBT1 small interference RNA against the K gene involved in the FASN and the CBT1 signaling, we knocked down the CBT1 gene using small interference RNA sequence for targeting the CBT1A messenger RNA. After incubation of the SKBR3 cells with a small interference RNA for 48 hours, this results in a significant more than 60% decrease in the CBT1 messenger RNA. As a consequence of that, we measure the messenger RNA expression for the other gene by RT-PCR, and after that silencing, the CBT1 downregulation resulted in a significant 40% decrease in ACC1 messenger RNA, and has no significant change for the expression of MCD. In contrast, CBT1A silencing in SKBR3 resulted in a significant more than 60% increase in the FASN messenger RNA expression, and after that, we test the apoptotic activity of CBT1 inhibition by an XM5, and as you can see, the incubation of the cells with the small interference RNA for the CBT1 lead to activation of apoptosis in the cells. Similarly, we test the effect of use ACC1A small interference RNA, as the acetyl-CoA carboxylase is a significant player in the pathway of fatty acid synthesis. As shown in the figure, as we can see here in the figure, transfection of the cells, I mean here the MDA231 cells with the ACC1 small interference RNA resulted in about significant 80% decrease in ACC1 messenger RNA. As a consequence of that, we test the messenger RNA expression of other gene by RT-PCR. The ACC1 silencing resulted in a significant about more than 50% decrease in FASN messenger RNA, and the same what we did for the previous cells. We test the effect of this inhibition on the apoptosis profile. As you can see from the flow picture, that this inhibition will lead to activation of apoptosis. After that, to examine the effect of using MCD small interference RNA on gene expression of the on the MCF7 cells with the MCD small interference RNA. After the incubation of the cells with the small interference RNA for 48 hours, this results in a significant around 70% decrease in the MCD messenger RNA expression. After that, we test this effect on the other genes by RT-PCR. We found that there is no much more significant. There is a little increase, but it's not significant for other cells. And after that, we test the effect of the apoptosis profile on the transfected cells, and we found that the treatment of the MCF7 cells with the MCD small interference RNA lead to 28% of cells suffered from early apoptosis, whereas around 50% of cells suffered from late apoptosis. Next, we test the effect of egenol on MDA, MB231, and SKBR3 cells to test the viability, and the result of proliferation assay indicate that egenol and with the concentration and a time-dependent manner effect on the MDA and SKBR3 cells, and this suggesting that there is a potential anti-proliferative and anti-apoptotic effect of egenol in both cell lines. And as we can see in the graph, at 40 and 60 micromolar of egenol, this will lead to inhibition of cell proliferation by more than 90% in both cell line. And this is the graph originated from the data of MTT. As you can see here, we test the cells for 24 hours and 48 hours with different concentration of egenol. Also, we did the same with the real-time cell proliferation using, we test the cell proliferation using the RTCA system. We monitor the cell for 72 hours to detect the real-time proliferation. We use three different concentration in the MDA cells and other 3 different concentration with the SKBR3 cells. The graph on the top represented the RTCA system. The one in the top, it's represent the lower dose. It's the DIMSU or the normal cells, the purple color, and the green color represents the 4 micromolar of phenol. The green color is the 8 micromolar of phenol, and the red color is 10 micromolar of phenol. Also, we did the same for the SKBR3 cells, and we detect that for 72 hours. After that, before that, it's important to mention that the IC50 that we have it from the MTT and the RTCA system is almost similar. According to the IC50, we choose 2 doses for the MCD. We use 4 micromolar and 8 micromolar, and for the SKBR we use 5 micromolar and 10 micromolar. After that, to investigate the possible effect of phenol treatment on the expression level of the fatty acid synthase in MDA and SKBR3 cells, after treatment for 48 hours with different concentration of phenol, we quantify the messenger RNA expression level by RT-PCR. Our data shows here that the use of phenol in MDA breast cancer lead to a significant increase in FASN gene expression. Interestingly, the treatment of SKBR3 breast cancer cells with 5 and 10 micromolar of phenol resulted in a dose-dependent significant increase in the messenger RNA expression of FASN as compared to DIMSO. Also, to investigate the effect of phenol treatment on the expression level of CPT1A in MDA and SKBR3 cell lines after treatment for 48 hours with different concentration of phenol, we quantify the CPT1 messenger RNA expression by RT-PCR. As shown here, treatment of MDA cells with 8 micromolar of phenol for 48 hours, this results in a significant around 80% inhibition in the messenger RNA expression of CPT1 as compared to the DIMSO-treated cells. Conversely, treatment of the SKBR3 cells, which is the HER2 positive cells, resulted, treatment these cells with a 5 and 10 micromolar of phenol for 48 hours resulted in a significant increase in the messenger RNA expression of CPT1A as compared to the DIMSO. Furthermore, we investigate the potential effect of using the different concentration of phenol on the messenger RNA expression of ACC1 in the cells by RT-PCR, and our results showed that the treatment of the cells for 48 hours resulted for 48 hours has no significant effect in the messenger RNA expression of the ACC1 as compared to the DIMSO. Conversely, treatment of the SKBR3 cells with phenol for 48 hours resulted in a significant 80% and 60% decrease in the messenger RNA expression of ACC1 in a respective manner as compared to DIMSO-treated cells. Also, we try to measure the effect of phenol on the MCD messenger RNA expression in the MCD and the SKBR3 cells by RT-PCR, and our results showed that the treatment of the cells, I mean here the MDA for 4 micromolar and 8 micromolar of phenol resulted in a significant more than 150% increase in the messenger RNA expression of MCD respectively as compared to the DIMSO. In contrast, a treatment of SKBR3 cells with the 5 and 10 micromolar of phenol resulted in a significant around more than 90% decrease in the messenger RNA expression of MCD compared to the control. After that, we try to test the wound. We try to test the effect of the phenol on the cell migration. We use the wound healing assay and the phenol with different concentration and different time point. And as you can see here from the figure, there is a significant inhibition for the cell migration, and this inhibition has a time dependent and concentration dependent on cell migration. We did the same for the SKBR3 cells, and the inhibition of the significant in cell migration is not that much, but we could not see any significant difference from the microscopic picture. But we go for a further investigation. Let's see where if there is and some involvement of the matrix metalloproteinases of the gene engaged on the tumor metastasis. We test the MMP2, MMP9, in the SKBR3 cells and MDA, and after the incubation with different concentration of egenol. And as you can see, that we have a significant inhibition of the MMP2 after different concentration of egenol in the two different cell lines in the term of messenger RNA and protein expression. Also, the data of matrix metalloproteinase 9 show a significant inhibition in both cell lines in the term of messenger RNA and protein expression. Also, we test the activity, the expression of TIM1, which is a tissue inhibitor of metalloproteinases 1, because this glycoprotein works as a natural inhibitor for matrix metalloproteinases, and we found there is no inhibitory effect for the egenol on the activity of TIM1 after we use the different concentration of egenol in the we test that in the messenger RNA and protein expression. We have no effect. Also, to explore the ability of the egenol to activate the caspases, we test here different caspases, and we test the ability of egenol to modulate the expression level of apoptotic gene caspase 3 in MD and SKBR3 cells after 48 hours of incubation. And as you can see, we found there is a significant increase in the level of caspase. After that, we test that in the messenger RNA. We use the normal caspase 3 and cleaved caspase 3, and the data from the messenger RNA show there is an active and high expression of the caspase 3. The same thing we did for the caspase 7, and the data for caspase 7 are highly expressed. The same thing also for caspase 9. The data from caspase 9 are highly expressed from the messenger RNA and protein expression. After that, we try to investigate the effect of egenol concentration on and the activation of those different apoptotic gene. We test the apoptotic activity by using the flow cytometry, as you can see in the left side. The control for the MDA and the SKBR3 cells, followed by different concentration of egenol. Once we have more concentration, this will lead to activate the apoptosis in the cells, and more of the cells go for apoptosis. Let's go for the conclusion of this work. I will start the first conclusion. FASN and CPT1 are differentially and highly expressed in SKBR3 cells, and targeting these genes is emerging as one of the most exciting and promising targeted therapy. One of the best strategy to treat HER2 breast cancer cell types. Sorry. ACC1 is highly expressed in MDA MP231 cells, and therefore triple negative will benefit more from ACC1 inhibition than another breast cancer subtypes. MCD show higher expression in MCF7 cells, and therefore it is an ideal therapeutic target in luminal A breast cancer subtypes. Also, the use of small interference RNA against FASN CPT1A, ACC, or MCD in breast cancer cell lead to cell apoptosis and considered as an ideal therapeutic target. Breast cancer treatment may need to be tailored to individual subtypes, and in fact, our results warrant a detailed mechanistic studies to monitor the exact level of acetyl-CoA, malonyl-CoA, and ceramide, as well as monitoring the protein expression codified by these genes. Also, the use of egenol in MDA and SKBR3 cells lead to cell apoptosis through modulation of FASN, CPT1A, ACC1, or MCD. And the present study demonstrated that the egenol exhibit an anti-breast cancer effect via targeting the caspase pathway, and may be considered as a potential therapeutic target for triple negative and HER2 positive breast cancer cells. As an output from this project, alhamdulillah, this project has been funded from CACS. We have 3 direct manuscripts originated from this a project. We have already one published manuscript in the PMC Complementary and Alternative Medicine. And we have one submitted manuscript, and the third manuscript is underwritten. The second manuscript, which has been submitted, the title of that manuscript is the agenol-induced autophagy and apoptosis in breast cancer cell line via inhibition of PI3 kinase AKT FOXO3 pathway. And the data for this manuscript not being included on the thesis or in this presentation. Also, we published two posters, and both of those two posters winner. For example, the first one earned the second place in the poster presentation in King Faisal Specialist Hospital and Research Center Breast Cancer Symposium, and the second one earned the first position in the CFA 2020 for the poster presentation for professional. By this, I reached the end of my presentation, and really I would like to thank you for your patience and for your time. But I have to acknowledge a couple of people and an institution. I would like to acknowledge King Saud University to give me the opportunity to complete my PhD in the great college and the great Department of Pharmacology and Toxicology. Also, I would like to thank King Abdulaziz Medical Center for Science and Technology for providing me with the fund for this project. Also, I would like to thank my institution, King Abdullah International Medical Research Center, to give me the opportunity and the time to finish my PhD. And also, excuse me, I will shift for Arabic because I feel it's more emotional than only science. اسمحوا لي أن قد أتقدم بشكر جزيل جدا للأستاذ الدكتور عثمان الشبانة مشرفي على وقته وجهده ومساعدته الدائمة وتوجيهاته. حقيقة لم يبخل علي بالنصيحة ولا بالجهد، يعني كان متابع لكل التفاصيل بدقة ومتابع للمشروع من أول لحظة وحتى آخر لحظة. يعني لا أجد كلمات توفي حقه في الشكر. أيضا أتقدم بشكر الجزيل والعرفان لأستاذي الأستاذ الدكتور محمد السيد مشرفي السابق. حقيقة تعب معي كثير في بداية هذا المشروع، وحتى بعد سفره كان يتواصل معي بشكل دائم ويتابع العمل. أتعبته معي كثيرا، وله جزيل الشكر والتقدير، ولم يتوانى عني في النصيحة في أي لحظة. أيضا أحب أن أتقدم بجزيل الشكر لمعلمي الدكتور محمد حافظ. حقيقة قضينا أيام جميلة وساعات طويلة جدا جدا في المعمل. تعلمت منه الكثير والكثير. تعلمت كيف تعمل في مجال الأبحاث، وكيف تبحث عن المعلومة، وكيف تتحمل الإحباط والفشل بعد الفشل في التجارب حتى تصل إلى نتيجة جيدة، فله مني كل الشكر والتقدير. أيضا أحب أن أتقدم بجزيل الشكر والعرفان لكافة أعضاء هيئة التدريس في قسم علم الأعضاء، قسم الفارماكولوجي والتوكسيكولوجي، قسم علم الأدوية والسموم، لكافة أعضاء التدريس، زملائي طلاب الدراسات العليا. حقيقة يعني لا أستطيع أن أذكر شخص واحد، لكن جميع أعضاء التدريس منهم من استفدت منه معلومة، منهم من نصحني بنصيحة، منهم من علمني على تقنية جديدة، منهم من تعلمت منه معلومة، فلهم مني كل الشكر والتقدير. وإن شاء الله سبحانه وتعالى أن التعاون سوف يكون مستمر وقائم في قادم الأيام بإذن الله. أيضا أحب أن أقدم جزيل الشكر والتقدير لأعضاء لجنة المناقشة على وقتهم وجهدهم وتعبهم لمراجعة الرسالة، وأنا متأكد بشكل كبير أن كل الملاحظات التي سوف يتقدمون بها لي سوف تكون محل التقدير والاهتمام، وسوف تؤدي بعون الله إلى منتج أفضل لهذه الرسالة. لا يفوتني في نهاية هذه الرسالة أن أشكركم على حسن حضوركم واستماعكم، وأيضا أتقدم بعظيم شكري وامتناني لأهلي، لوالدي ووالدتي وزوجتي وطفلتي على دعمهم وصبرهم على كل هذه السنوات، وشكرا لكم على استماعكم. وأتمنى إذا كان في لديكم أي استفسارات أو تساؤلات أنا سعيد جدا بتلقيها وشكرا لكم.